Welcome back to our fascinating journey through the world of biology. Today, we're delving into a tiny, yet incredibly vital organelle that plays a crucial role in your body's metabolism and detoxification. That's right, we're talking about the incredible world of peroxisomes. But wait, what exactly are peroxisomes, and why should you be intrigued by them? Well, my curious friends, that's what we're going to explore in today's video. In this video, we will unravel the mysteries of peroxisomes, from their discovery to their remarkable functions inside your cells. You'll learn how these tiny cellular powerhouses are involved in lipid metabolism, detoxification, and even the breakdown of fatty acids. Plus, we will uncover the secrets of peroxisomal targeting signals and how these organelles ensure your body stays in tip-top shape. So, whether you're a biology buff, a science enthusiast, or just someone curious about the inner workings of your own body, you won't want to miss this deep dive into the fascinating realm of peroxisomes. Stay tuned, because by the end of this video, you'll have a newfound appreciation for these tiny yet mighty organelles. So, without further ado, let's embark on our peroxisomal adventure together. Don't forget to hit that subscribe button and ring the notification bell so you never miss an exciting episode of our biology exploration. Now, let's start our journey into the incredible world of peroxisomes. Let's roll. What is peroxisome? Peroxisomes, integral components of eukaryotic cells, are membrane-bound organelles with a primary function in lipid metabolism and the detoxification of reactive oxygen species. These organelles play a pivotal role in the breakdown of lipids, particularly fats, which are energy-dense molecules. Given the hydrophobic nature of lipids, their metabolism in the aqueous cellular milieu presents challenges. Peroxisomes address this by providing a specialized environment for such metabolic processes. These organelles are equipped with oxidative enzymes that facilitate various metabolic reactions, including those related to energy metabolism. Notably, peroxisomes engage in the catabolism of specific fatty acids, such as very long-chain and branched-chain fatty acids. Additionally, they are involved in the synthesis of plasmalogens, ether phospholipids essential for the optimal functioning of mammalian brains and lungs. Another crucial function of peroxisomes is the management of reactive oxygen species. They are adept at converting potentially harmful molecules like hydrogen peroxide into benign substances, namely water and oxygen, thus safeguarding cellular integrity. The discovery of peroxisomes can be attributed to the Belgian cytologist Christian de Duve in 1967. These organelles were initially termed microbodies due to their minuscule size and vesicular structure. De Duve's research illuminated the presence of oxidases within peroxisomes that produce H2O2 and catalase enzymes that subsequently neutralize H2O2. Owing to their pivotal role in peroxide metabolism, the term peroxisomes was coined. Peroxisomes are especially prevalent in cells of detoxifying organs, such as the liver and kidneys. In the liver, they participate in the metabolism of bile acid intermediates and are instrumental in lipid storage, degradation, and synthesis. Their presence and function underscore their significance in cellular health and metabolic processes. Structure of peroxisomes. Peroxisomes are subcellular organelles, typically ranging from 0.1 to 1 micrometer in diameter, found ubiquitously in eukaryotic cells, encompassing both plant and animal cells. These organelles are characterized by their spherical shape and are encased by a single lipid-protein biomembrane. This membrane delineates the granular matrix from the cytoplasm, creating a specialized compartment that fosters a myriad of metabolic reactions essential for cellular function. The structural integrity of peroxisomes is maintained by a phospholipid bilayer, which is predominantly synthesized in the smooth endoplasmic reticulum. Embedded within this bilayer are numerous membrane-bound proteins. The enzymes crucial for lipid metabolism, which are housed within the peroxisome, are synthesized on free ribosomes in the cytosol. These enzymes are then selectively imported into the peroxisome, guided by specific signaling sequences, with peroxisome target sequence 1 being the most prevalent. Interestingly, the size, shape, and protein composition of peroxisomes can vary based on the cell type and its environmental conditions. For instance, in certain yeast species, the availability of glucose can influence the size and number of peroxisomes. When provided with an ample glucose supply, only a few small peroxisomes might be present. However, when exposed to long-chain fatty acids as the primary carbon source, the yeast may exhibit a significant increase in peroxisome size and number. It's noteworthy that peroxisomes do not possess their own DNA. Instead, the proteins they house are translated in the cytosol and subsequently transported into the organelle. As proteins and lipids are continually incorporated, the peroxisome enlarges and eventually divides, yielding to distinct organelles. This dynamic nature underscores the adaptability and essential role of peroxisomes in cellular metabolism. Peroxisomal enzymes. Peroxisomes are replete with a diverse array of enzymes, with approximately 60 distinct enzymes identified within their matrix. These enzymes play a pivotal role in facilitating oxidation reactions, culminating in the generation of hydrogen peroxide. Among the primary groups of enzymes housed within peroxisomes are urate oxidase. This enzyme is instrumental in the oxidative breakdown of urate, a process integral to purine metabolism. Diamino acid oxidase. This particular enzyme is responsible for the oxidation of diamino acids, contributing to amino acid metabolism. Catalase, a crucial enzyme, catalase acts to decompose hydrogen peroxide into water and oxygen, thereby mitigating the potential cytotoxic effects of hydrogen peroxide within the cell. These enzymes underscore the essential metabolic functions of peroxisomes, emphasizing their significance in cellular biochemistry and homeostasis. Peroxisome biogenesis. Peroxisomes are vital cellular organelles that play a pivotal role in various metabolic functions. The efficacy of these functions is intrinsically linked to the accurate biogenesis of peroxisomes and the proteins they contain. Unlike other organelles, peroxisomes do not possess nucleic acids. Instead, all proteins found in peroxisomes are encoded by nuclear genes and are synthesized in the cytoplasm. These proteins are then post-translationally imported into the peroxisome. Peroxisomal membrane proteins, PMPs, and their biogenesis. PMPs are integral to the peroxisome membrane and are characterized by cis-acting peroxisomal targeting signals, known as MPTSs. These signals are multipartite and encompass at least one transmembrane domain and a binding site for PEX19, a chaperone and import receptor for PMPs. PEX19 plays a dual role. It binds to newly formed PMPs in the cytoplasm, ensuring their solubility, and subsequently transports them to the peroxisome's outer surface by interacting with PEX3. The exact mechanism of PMP import post this step remains elusive, but it is believed to involve the detachment of PMPs from PEX19 and their integration into the peroxisome membrane. Peroxisomal matrix proteins and their biogenesis. In contrast to PMPs, peroxisomal matrix proteins, which encompass the majority of peroxisomal enzymes, need to traverse the peroxisome membrane. These proteins are equipped with specific peroxisomal targeting signals, PTSs, either PTS1 or PTS2. PTS1 is a tripeptide located at the C-terminus, while PTS2 is a nonapeptide situated near the N-terminus. These signals are recognized and bound by their respective receptors, PX5 for PTS1 and PX7 for PTS2. These receptors then transport the enzymes to the peroxisome, facilitated by the docking factor PX14. The import process involves several other peroxins that aid in the translocation of these enzymes into the peroxisome and the subsequent recycling of the receptors. Diseases linked to peroxisome biogenesis. Defects in the genes responsible for PMP or peroxisomal matrix protein import can lead to Zellweger syndrome, a severe inherited disorder marked by a complete loss of peroxisomal metabolic functions and various neurological, hepatic, and renal abnormalities. Mutations in PEX7 result in a distinct disorder known as rhizomelic chondrodysplasia punctata, RCDP, type 1, characterized by specific defects in peroxisomal functions. Peroxisome division and formation. Peroxisomes predominantly form through the budding or scission of existing peroxisomes. This process involves peroxins from the PEX11 family and the GTPase DLP1. However, under certain conditions, peroxisomes can also form de novo. This process, observed under specific laboratory conditions, is believed to involve vesicle release from the endoplasmic reticulum, that matures into functional peroxisomes. Division and proliferation of peroxisomes. Peroxisomes, vital cellular organelles, undergo intricate division and proliferation processes. Two primary models have been proposed to elucidate the division of peroxisomes. The first model suggests that peroxisomes originate by budding and subsequent fission from pre-existing peroxisomes, a mechanism potentially predominant in mitotically dividing cells. The alternative model posits that peroxisomes emerge either de novo or from a distinct membrane reservoir, possibly the endoplasmic reticulum, This reservoir then undergoes a series of biogenesis intermediates to form mature peroxisomes. This latter model might be more relevant to proliferative peroxisomes or the regeneration of peroxisomes in PEX mutants upon gene complementation. In the yeast species, Yarovia lipolytica, the initiation of peroxisome division is internally signaled. This signal might be generated through the remodeling of peroxisomal membrane lipids or via metabolites produced by peroxisomal fatty acid beta oxidation. To understand the intricacies of peroxisome biogenesis, several proteins play pivotal roles. Proteins facilitating peroxisome formation from the PEX3P, an integral peroxisomal membrane protein, PMP, conserved across species, essential for the assembly and stability of the ring domain subcomplex. PEX16P, found in Y. lipolytica. This protein aids in the import of specific peroxisomal proteins and inhibits peroxisome division. PEX19P, a farnesylated protein that acts as a chaperone and import factor for newly synthesized integral PMPs. Matrix protein import proteins, PEX1P, an ATPase involved in peroxisome targeting signal, PTS, receptor recycling during import. PEX2P, an integral PMP with a ring domain, involved in receptor recycling. PEX5P, the primary receptor for PTS1, interacting with multiple peroxins. PEX6P, an ATPase that collaborates with PEX1P for PTS receptor recycling. Proteins regulating peroxisome size and number. PEX11P, essential for maintaining normal peroxisome abundance and division. PEX25P, PEX27P, PEX28P, PEX29P. These proteins play roles in modulating peroxisome size, number, and distribution. Peroxisome inheritance proteins, INP1P and INP2P, fungal PMPs that play roles in peroxisome retention and movement, respectively. Myo2P, an actin-based motor protein facilitating peroxisome movement. In essence, the division and proliferation of peroxisomes are orchestrated by a complex interplay of proteins, ensuring the proper functioning and distribution of these organelles within the cell. Peroxisomal fatty acid bioxidation. Peroxisomal fatty acid beta oxidation is a conserved metabolic process observed across a diverse range of organisms, from yeast and plants to invertebrates and vertebrates. Notably, while yeast and plants rely solely on the peroxisomal pathway for fatty acid beta oxidation, lacking a mitochondrial counterpart, vertebrates possess both peroxisomal and mitochondrial pathways. The core process of peroxisomal fatty acid beta oxidation comprises four fundamental steps. Acyl-CoA oxidation. The initial step involves the oxidation of fatty acyl-CoA to form to enol-CoA. This reaction is facilitated by acyl-CoA oxidase, which simultaneously reduces molecular oxygen to produce hydrogen peroxide. This distinctive feature differentiates the peroxisomal pathway from the mitochondrial counterpart, with the latter conserving the energy from acyl-CoA dehydrogenation for ATP synthesis. Hydration of 2-enol-CoAs. The next phase sees the conversion of 2-enol-CoAs to 3-hydroxyacyl-CoAs, a reaction catalyzed by enol-CoA hydratase. In peroxisomes, this enzyme often exists as part of multifunctional proteins, such as the dibifunctional protein. Dehydrogenation of 3-hydroxyacyl-CoA. This step involves the NUD-dependent dehydrogenation of 3-hydroxyacyl-CoAs, producing 3-ketoacyl-CoA and NAD. Thiolase-catalyzed cleavage. The final step involves the cleavage of 3-ketoacyl-CoAs by thiolase, resulting in the release of acetyl-CoA or propionyl-CoA for certain branched-chain fatty acids. Beyond these core reactions, peroxisomes house several auxiliary proteins essential for fatty acid oxidation. Metabolite transporters. These proteins, such as the PXA12 in yeast and ABCD1/4 in humans, facilitate the transport of acyl-CoA molecules from the cytoplasm into the peroxisome. Acyl-CoA synthetases. These enzymes activate specific free fatty acids to their acyl-CoA derivatives. ATP-ADP carriers. These support the intraperoxisomal activation of certain fatty acids. Enzymes for unsaturated fatty acid oxidation. These include 3-enol-CoA isomerase and dienol-CoA reductase, which aid in the oxidation of unsaturated fatty acids. Racemase activities. These convert certain branched-chain fatty acids to forms that can be degraded by peroxisomal enzymes. Acetyl-CoA export mechanisms. These transport acetyl groups from the peroxisome to the cytoplasm, a prerequisite for their mitochondrial oxidation. Metabolite shuttles. These regenerate the required NAD and transport reduced electron carriers out of the peroxisome. In mammals, the peroxisomal fatty acid beta oxidation pathway primarily oxidizes very long-chain fatty acids, bile acid precursors, and certain branched-chain fatty acids. In contrast, in yeast and plants, this pathway is the sole mechanism for converting fatty acids into acetyl-CoA, playing a pivotal bioenergetic role, especially during specific growth phases or conditions. Peroxisomal contributions to bile acid synthesis. Peroxisomes play a pivotal role in the metabolism of specific substrates, notably the cholesterol-derived bile acid precursors, dihydroxycholestanoic acid and tridroxycholestanoic acid. These substrates are unique in that they are exclusively metabolized by the peroxisomal beta oxidation enzymes. DHCA and THCA, characterized as large, lipophilic, 2-methyl branched-chain fatty acids, undergo a series of metabolic transformations within the peroxisome. Initially, these fatty acids are esterified with coenzyme A. Following esterification, they are transported into the peroxisomal matrix. Within this matrix, they undergo beta oxidation, resulting in the production of propionyl-CoA and cholyl-CoA. Furthermore, peroxisomes house a specific enzyme known as bile acyl-CoA, amino acid acyltransferase. This enzyme facilitates the transfer of the cholic acid moiety onto amino acids, specifically taurine or glycine. This enzymatic action culminates in the formation of bile acids. These newly synthesized bile acids are then transported out of the peroxisome, entering the cytoplasm. From the cytoplasm, they are directed into the bile duct, eventually reaching the digestive tract. In essence, peroxisomes are integral to the synthesis and subsequent transport of bile acids, underscoring their significance in cholesterol metabolism and overall lipid homeostasis. Peroxisomal fatty acid oxidation. In the realm of cellular metabolism, both peroxisomal and mitochondrial beta oxidation systems collaboratively enable animals to metabolize a vast array of dietary fatty acids. However, specific dietary fatty acids, notably 3-methyl substituted ones such as phytanic acid, present resistance to beta oxidation. Despite this resistance, animal cells proficiently oxidize these fatty acids, primarily due to two distinct pathways: the unique peroxisomal fatty acid alpha oxidation pathway, which truncates phytanic acid by a single carbon, resulting in the formation of the 2-methyl branch-chain fatty acid, pristanic acid. The combined efforts of the peroxisomal and mitochondrial fatty acid beta oxidation pathways, which further degrade pristanic acid into acetyl-CoA and propionyl-CoA units. The fatty acid alpha oxidation pathway is a complex, multi-step process orchestrated by 4 pivotal peroxisomal enzymes. Activation of phytanic acid. Phytanic acid undergoes activation to form phytanol-CoA, facilitated by a peroxisome-associated enzyme known as phytanol-CoA synthetase. The precise location of this enzyme remains a topic of discussion. If it resides on the peroxisome's outer surface, a metabolite transporter, potentially from the ABCD1-4 family, might also be instrumental in the alpha oxidation process. Conversion by dioxygenase. Within the peroxisomal matrix, the enzyme phytanol-CoA to hydroxylase, a type of dioxygenase, transforms phytanol-CoA into 2-hydroxyphytanol-CoA. This reaction necessitates the presence of 2-oxobutyrate and oxygen. Degradation by lyase. The enzyme 2-hydroxyphytanol-CoA lyase degrades to hydroxyphytanol-CoA, releasing pristanyl and formyl-CoA. The latter spontaneously disintegrates, producing free CoA and formate, which eventually gets released as CO2. Oxidation of pristanyl. Pristanyl undergoes oxidation via pristanyl dehydrogenase, an NAD-dependent process, resulting in pristanic acid. This acid is then activated to pristanol-CoA, which subsequently undergoes peroxisomal fatty acid beta oxidation. It's noteworthy that the mammalian peroxisomal beta oxidation pathway only partially truncates its substrates. This is evident in the oxidation of pristanol-CoA, where 3 rounds of peroxisomal beta oxidation produce acetyl-CoA to propionyl-CoA molecules and the branch-chain fatty acid 4,8-dimethylenol-CoA. This fatty acid is then exported from the peroxisome for further mitochondrial oxidation. The exact mechanism of this export, whether through free fatty acid, a carnitine ester, or a combination of both, remains to be elucidated. Ether phospholipid biosynthesis. Phospholipids, integral components of cellular membranes, predominantly poses a classic structure grounded on glycerol 3-phosphate esterified with fatty acids at its 1 and 2 carbons. Yet, a notable fraction, approximately 15–20% of phospholipids in humans, deviates from this norm. These unique phospholipids feature a fatty alcohol linked to the 1 carbon via an ether bond. The synthesis of these ether-linked phospholipids is orchestrated by a series of enzymes distributed across the peroxisome, cytoplasm, and endoplasmic reticulum. The foundational steps of this biosynthetic pathway are rooted in the peroxisome, where enzymes facilitate the production of alkyl dehydroxycetone phosphate, alkyl DHA, the pivotal precursor for ether phospholipid synthesis. The primary stages encompass glycerol 3-phosphate oxidation, a peroxisomal enzyme, glycerol 3-phosphate dehydrogenase, facilitates the conversion of glycerol 3-phosphate into DHA. Formation of 1-acyl DHAP. Within the peroxisome, DHAP acyltransferase mediates the synthesis of 1-acyl DHAP, utilizing DHAP and a fatty acyl-CoA. The resultant acyl DHAP is a crucial substrate for subsequent alkyl DHAP synthesis. Reduction of acyl-CoAs, situated on the peroxisome's exterior. The NAF-dependent fatty acyl-CoA reductase transforms acyl-CoAs into fatty alcohols. These fatty alcohols serve as essential substrates for alkyl DHAP production. Synthesis of 1-alkyl DHAP. The enzyme alkyl dihydroxyketone phosphate synthase, residing within the peroxisome, orchestrates the formation of 1-alkyl DHAP. This reaction involves 1-acyl DHAP and a fatty alcohol, simultaneously liberating a free fatty acid. Formation of 1-alkyl G3P. The alkyl DHAP reductase, present on the cytoplasmic facet of both peroxisomes and the ER, oversees the NUT-dependent synthesis of 1-alkyl glycerol 3-phosphate, 1-alkyl G3P. Subsequent functions of peroxisomes. Peroxisomes, integral organelles within eukaryotic cells, perform a myriad of vital functions that are indispensable for cellular homeostasis and metabolism. These functions include hydrogen peroxide metabolism. Peroxisomes house enzymes that are pivotal for both the generation and detoxification of hydrogen peroxide, H2O2, a reactive oxygen species. This dual role ensures that while H2O2 is produced for specific metabolic reactions, its accumulation is kept in check to prevent cellular damage. Fatty acid oxidation. Peroxisomes play a central role in the oxidation of fatty acids. In animal cells, this process transpires in both peroxisomes and mitochondria. However, in plants and yeasts, it is predominantly a peroxisomal function. The oxidation process is concomitant with H2O2 production, which is subsequently neutralized by the catalase enzyme, underscoring the organelle's role in energy metabolism. Lipid biosynthesis. Peroxisomes, in conjunction with the endoplasmic reticulum, are involved in the synthesis of lipids such as cholesterol and dolitol. In the liver, peroxisomes facilitate the conversion of cholesterol into bile acids. Additionally, they harbor enzymes essential for the synthesis of plasmalogens, phospholipids that are crucial for the proper functioning of cardiac and neural tissues. Germination of seeds. During seed germination, peroxisomes are responsible for converting stored fatty acids into carbohydrates. This transformation is vital, providing the necessary energy and substrates for the burgeoning growth of the germinating plant. Photorespiration. In leaves, especially green ones, peroxisomes collaborate with chloroplasts to execute the process of photorespiration, a metabolic pathway that operates when the concentration of carbon dioxide is low. Degradation of purines. Peroxisomes are involved in the catabolism of molecules such as purines, polyamines, and certain amino acids. Enzymes like uric acid oxidase play a significant role in this degradation process. Bioluminescence. In fireflies, peroxisomes contain the enzyme luciferase, which facilitates bioluminescence. This luminescent property aids fireflies in tasks such as mate selection and prey capture. In summary, peroxisomes are multifunctional organelles that contribute significantly to various metabolic pathways, ensuring the proper functioning and survival of eukaryotic cells. Thank you for watching this video. You can get the study notes related to this lecture video from our website. Go to Google and search microbiologynode.com and you will get the link of our website.